So you're going to be seeing a presentation from the reading course Analyzing Ecological Integrity, Assessing Princeton University's Natural Areas. Before we dive into things, I want to first send a big shout out to Shauna Weber, Caroline Savage, and Gina Talt at the Office of Sustainability, as well as all the folks at Facilities and Sustainability that helped us out with this project. Especially a big thank you to Professors Adam Maloof and Amanda Irwin-Wilkins for all of your hard work in helping the class move along. We're going to start today talking about two words, baselines and reproducibility. And these are going to be themes that you're going to see through the entire talk today. And what we really mean by this is, I'm going to explain through a short analogy. So imagine knowing the time that you finished a track race, but not knowing the time at which you started, right? Without a baseline, how do you measure your success and how do you measure your improvement? Or similarly, imagine comparing race times between different races that you've run, but of races of different unknown lengths, right? So how do you compare different metrics that are being measured when they're measuring different things, or you're not sure exactly what they're measuring, right? So that has to do with reproducibility. And if you can't reproduce your results every time, or if you can't reproduce previous results that have been collected, and you don't have a baseline, how can you measure the success of a project? How can you measure improvement without knowledge of initial conditions and without clear and reliable measurement methods? And this is really what we're going to be focusing on today in the context of the Princeton campus ecosystem. And so I'm going to start by kind of taking a long view of the Princeton campus baseline and using historical land use as the context for which ecology and sustainability efforts are occurring. And this is sort of a qualitative overview of how campus has changed from 1875 to today. You can see lots of development, obviously, and as well as where it's headed in the future. More and more development, and so more and more of this strong interaction between the human use and the natural processes. But we really want to take a much more specific look at where things have been changing and where things haven't. So what you're going to see now is a montage of different drawings that were done by a consultant firm from historical maps and lithographs. And these are really helpful at looking to see generally how campus has changed over the past several hundred years. So this is the first year Princeton University was in town. There are two buildings that you can see, mostly just cleared land. 1852, we've already got the Delaware and Raritan Canal that's been built, a pretty significant change in land use. We've got more and more expansion towards the beginning of the 20th century. 1906, the construction of Lake Carnegie Dam vastly changes how land is being used here. In the 70s, more and more expansion until finally, most recently, we've seen increasing expansion and development in West Windsor, which ultimately is where the campus plan is headed, to more and more development on that side. And these pictures are really good at capturing an immediate visual for where things are changing and how different land is being used. You can see open areas, you can see wooded areas, developed areas. But what's important to note is that these maps, especially the really early ones from the colonial era even, these are mostly qualitative maps based on drawings and hand-drawn maps from the past, and it's not immediately evident where all of these sort of land classifications came from, especially, as I said, some of the older ones. And to really be able to use this as a strong tool in comparing how land change has affected other measurable like natural changes, such as water quality, such as the trees and fauna that we see in biohabitat areas. We want to be able to put very exact numbers and locations with low error on each of these small little patches to be able to track them through time. And to do that, we took a look at historical photography, which we had access to going back to 1940. And we're going to pick out the land uses that we really care about: developed land, cleared land, and forested land, to really get a picture of how campus is changing. So I'll walk you through that process. We start with a picture, for example, in 1951, that we can orient in space, and you can see here the campus border around the area that we care about, and we can then zoom in and draw individual lines around the parts of the campus that we want to be able to measure. So, for instance, there's the polygon for the water. We can draw polygons by hand over each forest patch. We can then fill in all of the cleared areas, and then on top of that fill in all of the developed areas. And what's important to note is that these are very similar to the last figures that you saw, but because these are developed in mapping program and not just the PDF of the picture, we can put exact areas on each of these things that we know to be highly accurate from the historical photography that we took it from. And what's really important about this is that we can take this and do it for every year and watch how those patches in different areas have shrunk and grown. So here we have 1940 to 2016 for the years for which we had access, and you can see here the cleared lakeshore in 1940 over there is now much more forested. On the West Windsor side, you can see development of various grad colleges in Alexander Road. Most recently, the solar field that was put in the 2000s, and the clearing of the Butler Tract. And we can track these changes through time, and we can also, using similar methods, project how will future expansion change land use and affect different areas on campus. And so to visualize that in a different way, we're going to look at a graph. You're going to see years on the bottom, and then the y-axis you're going to see area in absolute terms in kilometers squared, and area as a percentage of campus. And so this is the development of campus, the area that has buildings. So an increase since 1940 of about 1 square kilometer, and this land came from land mostly that was previously cleared. And what we see here is a small increase in the amount of trees on campus. And so we can ask, okay, so where are these trees, where is this forested area being added? And what you can visualize in the bottom here, West Windsor is the dark green, main campus is on the Princeton side is the light green. You can see that main campus forest patches have taken a slight dip while West Windsor has increased dramatically. 32% shift in the amount of total forest that is now in West Windsor as opposed to Princeton from 1951. And what's important to note here is that although it is a positive thing ecologically for forest growth to be occurring, increasing the total forest area does not necessarily mean increasing quality directly, as older and mature trees are much more valuable in terms of being able to support wildlife, in terms of being able to keep endangered species at bay. And so the newer forest on West Windsor is much more vulnerable, which is especially important when we consider where campus is going. So this is the line of about where we are now in 2017. And this is where you're going to see that development. So most of the development, a lot of it is in the cleared land in West Windsor, or an already cleared land on campus. But as you saw in the last map, it's right up against those new forest patches on the West Windsor side of more vulnerable forest. So I took you through sort of about 80 years of Princeton land development through historical photography, and now we're going to have a few of our other groups sort of zoom in and give you vignettes of field work looking at very specific ecological problems on campus.
Okay, so for context, like Benji said, the next two presentations are both going to be examples of zooming in on one specific aspect of ecology, stream health and forests. What Alec and I are interested in is seeing if we can use sedimentation in Lake Carnegie as a proxy for basically an integrated measure of all the activity over all of campus over time. So these are two images that you saw from Benji, but I'm zooming in. So that's Jadwin Jim to orient you. And what you see here is that adjacent to Lake Carnegie, going from 1940 to present, there's a dramatic change in land use right adjacent to the lake. We go from a time where there's no tree cover to pretty forested areas. And we're interested in how this affects sedimentation in the lake. So the basic model is that you have all of campus above here draining through erosion, sediment that's fluxing through these rivers and drainages and ending up in the lake and building up through layers of sedimentation. Now we can think that if you go from 1940 to today, how the land use right adjacent to the lake and how the rest of campus has affected the rates at which sedimentation is coming. So you can imagine a slower sedimentation rate. We're interested in basically quantifying these two arrows. One, how has the sediment flux from Princeton's campus changed over time? And two, how does the land use right adjacent to the lake affect sedimentation rates?
So to answer these questions, we conducted a survey of Lake Carnegie, in which we measured water depths along the same transects that a 2013 Princeton Hydro survey did. Here were our tools. We had a biosonics depth measurement, which uses it's an echo sounder that uses sonar to basically measure the distance between the instrument and the bottom of the sediment. We used a GPS, and we put it all in a canoe and paddled around the lake. And here you can see our transects in red, and the blue transects done by Princeton Hydro in 2013 in blue. And so what we did was we measured the difference between water depths between our survey in 2017 and theirs in 2013. So you can see here is that first, this transect followed their transect almost perfectly. And what that means is that the methods are reproducible. We could do exactly what they did without any problems. And their data, seen here in blue, can serve as a baseline from which you can measure change in water depth over time. So it seems perfect. However, that change in water depth over time, that offset between the blue line and the red line, is the result of an aggregate effect of real sedimentation, which we want to measure, and a uniform, unknown offset in absolute water level that is the result of changing water levels by draining the canal or the lake by the dam. We don't have that measurement of absolute water level. We were not able to get it, and so we don't know exactly, we can't pin an absolute change to our thing because our baseline is unknown. To kind of overcome this challenge, we used a relative baseline. So instead of measuring an absolute change in sedimentation, we measured a change in sedimentation relative to the mean loss or gain of sedimentation. We did that by taking the mean of the offsets for all of our transects and basically offsetting our measurements by that mean.
Right, so now we can see a map of the relative change in sediment across the lake through time. So everything that's in green is places where sediment has accumulated the most, and places in red are places of relative erosion. And what you notice is everywhere where we have drainages coming off from Princeton, either streams or man-made drainages, you can see dramatic increase in sediment. And that's really exactly what we expect. These are the places where erosion is the streams are carrying sediment into the lake. But in order to turn these into measurements where we can say, for example, the number of millimeters per year of sediment, we need to have an absolute baseline. So sort of to put this in context, there's really a rich history of data for Lake Carnegie in the past 100 years. And you can see right off the bat, there's some really interesting trends. So for example, in the first half of Lake Carnegie's history, the lake had to get dredged 3 times. And in the past 50 years, it's never been dredged. And that makes you wonder, has there been something that has changed in the rate at which it's been filling up? And how is that related to land use on Princeton's campus? So our vision for the project was to correlate changes in land use like you just saw from aerial imagery from Benji to these data that show bathymetric constraints on Lake Carnegie. So these are just a handful of some of the data that exist. These two are the ones that we use. This is our survey, and this is data that Princeton Hydro was generous enough to provide us. Princeton didn't have the raw data. But unfortunately, our requests to access all of these things were denied. And then the last point is that in order to make future measurements on Lake Carnegie, all we really need to do is have some lake measure, some water level gauge, and that could be as simple as having a ruler or a PVC pipe with water depth markings installed on some dock on Lake Carnegie, so that every time people take a measurement on Lake Carnegie, they can refer to this absolute datum.
So we now have a better idea of what is going on with Lake Carnegie. But Lake Carnegie it's not an isolated system. Especially on and close to campus, there are 3 streams that feed into Lake Carnegie. And I want to draw your attention to this part of the campus extension plan, that the residential building south of Poe Field partly encroach on this area, which is very close to this stream, which we will call later tennis court stream. So the reason we care about these forested areas that are close to streams is because forested areas close to streams act as buffers against water pollution and thus help improve water quality. And here the way we want to think about water quality simply as a measure of the suitability of using this water for humans and also for ecosystem health. So how do exactly forested areas affect water quality? Basically what happens is that pollutants, which could be actual pollutants, so toxins, or also nitrogen and phosphorus in high concentrations, if there's no forest, or if it's developed land, for example, they just continue on their way, probably into Lake Carnegie. On the other hand, if you have a forested area, these pollutants can be taken up in the soil and maybe by the plants, especially in the case of nitrogen and phosphorus. So the concentration of pollutants is then decreased.
Now that we've just heard about how forested areas affect water quality, now we get into how we measure water quality. So there's lots of different metrics that we can use. It would be really exciting if we could have measured all of these things, but given budget and time constraints of one semester, we chose to focus on pH and conductivity since they're inexpensive and allow easily for long-term monitoring. So getting into what is pH. pH is on a scale from 0 to 14. 7 is the center of the scale, that's where neutral pH is, and that's where we notice that tap water ideally should be at. One thing I want to point out is that rainwater pH is much lower, it's at a pH of 5.5. Now, this may not seem like much of a difference because it's a 1.5 unit change. However, since pH is on a log scale, even a 1 unit decrease in pH means a 10 times increase in acidity. So later what we want to focus on is bringing your attention to this pH area. This is the healthy pH area according to the New Jersey State Water Guidelines from 6.5 to 8.5. If we go outside this range, for instance, if we go to the acidic range of the scale, then metals become more toxic. Obviously this has harmful effect on organisms. And similarly, if you go to the basic side of the scale, then that means you might have too high concentration of nutrients, which is also not beneficial to the organisms. So now going into conductivity, which is the second water quality measure that we use. Conductivity basically is a measure of how many ions there are in the water. So if you have more ions, you're going to have a higher conductivity. This usually means that there's also more dissolved solids, which means you'll have murkier water that doesn't look as aesthetically pleasing. And similarly to pH, if you have too high of a conductivity, you're also going to have negative effects on wildlife.
So this is a map of where we put all of our probes. So we decided to survey 3 different streams. Starting from the left, we have the golf course stream, which goes through the golf course first, and then through a forested area at the bottom. So there were 3 probes on that stream. Then we have the tennis court stream, which I presented at the beginning, which has 2 probes, one at the beginning and one at the end. And finally, we put 2 other probes on the Washington Road stream, which here we call the free chemistry stream. And finally, we had one last probe, which is not shown here, on Lake Carnegie. So this is how we conducted our study. We put PVC pipes anchored into the ground with metal rebar. And then we had 2 PVC pipes going into the water one with a conductivity probe and the other with a pH probe. And we drill hole into PVC pipes to allow water to flow through the pipe. Later in the study, we had to add foam floats to prevent the probes from drowning, as they are not completely waterproof. And so the idea is that the setup is still there now. We had to take out the probes because we didn't want them to freeze, but now students can go back and put back the probes and continue collecting data. And this is exciting because, I mean, this study is basically the baseline in this case, because we were able to access single data points collected every 3 months, but this is actually the only data we found, at least, that has like continuous data of pH and conductivity through time. So we collected data every 30 minutes, to give you an idea. And so this is what it looks like in real life. So this is a setup for tennis courts too. So these are our results for pH when there were no precipitation. So we call this dry weather. And so this is these 3 graphs represent streams with the probes upstream to the left. So FL1, FL2, TC1, and TC2. So FL is the free chemistry lab. It's the Washington Road. Tennis courts, tennis court streams, and finally the golf course there, again, upstream to downstream. And this represents Lake Carnegie. So we have pH shown on the box plot. So the line represents the median. And then the further you go away, the further you are from the median, with points here represent outliers. So value very far away from the median. And finally, we have the box here, which represents a healthy pH that we defined earlier. And so what you can see is that, for example, between TC1 and TC2 and GC2 and GC3, you have forested areas. And you see that between these probes, the pH was much more stable downstream after the forested areas. So you see here that this was, a lot of the data points here were below this healthy pH. And in TC2, it was actually stabilized, so it was much more stable, and it was also higher. Same thing in the golf course stream between the second and the third probe. You do not observe this, for example, between the first probe in the golf course and the second probe in the golf course. And this is because between these two, it's actually just a golf course, so no forests. Similarly, you don't see that big of a change for the Frick Chemistry Lab, so the Washington Road Lab. And this might be simply because originally the pH was already quite healthy on the first graph. In addition, there was another pipe here shown in black that might have made the data might have impacted the data. And finally, you can see that the Lake Carnegie here is very stable, which makes sense as it is a much bigger body of water.
So this is in sharp contrast to what we observe under wet weather. So I would in particular like to draw your attention to the Washington Road stream data and the golf course stream data. Under wet weather, what we observed, unlike what we observed under dry weather, is that moving from upstream to downstream, instead of seeing stabilization of the pH data, we see a much wider range in pH values, and we also see that the median pH has dropped considerably. It makes sense that the pH drops, given that rain pH is much lower than the average pH at 5.6. Similarly, when we go to the golf course stream, we also note, whereas it was very stable at the first probe, after precipitation, once you get further downstream, the median pH has dropped and variability has widely increased. However, going back to the lake is very stable. It's a much bigger body of water. However, what these results suggest is that even though we are observing the buffer effects of forested areas under dry weather, they are too small or fragmented to be able to effectively buffer extreme values of pH under wet weather, which is concerning because who we're going to hear from next are the people who actually studied the health of these forested areas, and we'll tell you more about them.
So as we just heard, forests are very important under dry conditions to help buffer the pH from entering streams. And forests are also very important for our campus in general. Not only do they provide recreation opportunities for students and faculty, they're also important wildlife habitat and have a lot of environmental benefits as well. And so to protect our forests, the Office of Sustainability, as well as the Office of the University Architect, in conjunction with some outside consulting firms, created a index to measure the relative value of all the forested areas on campus. And that's called the Playa Index for Land Use and Ecological Assessment. And so sort of here are the 18 metrics that make up this Playa Index. And you can see in the map on the right, this is one of the initial maps of the Playa metrics, and you can see going from low value in red to high value in green, all the different forest patches are given a single score as a composite of these 18 metrics. And so, in this study, we chose to focus on a limited number of metrics given our limited amount of time and our limited technical capabilities and equipment. And so we focused on 2 forest patches, Area C and Area F, known as the tennis court stream and the Frick Lab stream areas from the previous presentation. And in Area C and Area F, we measured 4 of the Playa metrics, and we also measured an additional 2 in Area C. We also measured one more metric that is not included in the Playa Index, and that is the Floristic Quality Index. This is a measure of vegetation quality, and we sought to understand Area C using that metric as well. So we tried to use the Playa document to base our methodology off of, so that we could be collecting the similar type of data that had already been collected to inform this document. However, on many of the metrics we were assessing, there was incomplete methods listed in this document. For example, here is a metric 10, invasive percentage understory, and so when we were looking at this, we wanted to implement some of the same methodology. However, there are many questions that were left unanswered, and we wanted to make sure that we had a very thorough baseline of methodology so that it could be reproduced in the future. So we decided that we needed to answer a lot of these additional questions so that we could have a methodology that could be repeated in the future.
So, here I've outlined the basic ideas of the motivation for having reproducible methodology as it relates to our studying these forested areas. And I'm going to touch specifically on these bottom dimensions here. First is the inter-patch variability that you can see in this map that you saw earlier. These numbers are based on those Playa metrics, which, as we've noted, are not super reproducible, and we don't really know the accuracy of those metrics. Unfortunately, in our study, we weren't able to perform investigations at that scale. However, concerning intra-patch variability, which you can see an example of here, as you can see, these are the number of large trees we saw just in Area C. At this scale, we will be able to show you both the establishment of a baseline as well as reproducible methods. And moving on to temporal data, and that's going to have a lot more to do with long-term monitoring techniques, and that's with regards to the way that the forest patches on campus are evolving naturally through time, as well as the way that they respond to disturbances and developments on campus. And as you can see shown here from the University Campus Plan for 2026, there's a proposed development here that's going to encroach just a little bit on the east side of Area C.
So for our methodology, in Area C and Area F, we measured all of the trees along these 2 transects in Area C, each 10 meters wide, as well as 4 10 by 10 meter boxes in Area F. For measuring the forest canopy, invasive species, and native diversity, we focus on a smaller scale. So in each of these boxes, which are 10 by 10 meters, we measured 5 different locations of 1 by 1 meter size. And I'll explain a little bit more how we measured forest canopy in a minute. We also measured the light levels and sound levels at 4 locations along our first transect of Area C to try and understand how they varied within the patch. And as I was saying earlier, for measuring the percentage of canopy, we utilized a really rich data set that is managed by the facilities. And this is called LiDAR. And LiDAR is a type of remote sensing, like Benji was explaining in the beginning. However, instead of returning an image, it has LiDAR returns, which are points in space. And you can see that here. Here is a sort of cross-section of the forest. You can see here's a tree coming up, and there's some canopy up above. And so we use that to calculate the percentage of returns that were above 22.5 meters, which was our cutoff for the canopy height.
So next I'm going to show you some of our results from Area C.
here's that map again that I showed you just earlier with all of our large trees in the area. And I'm going to tell you more about that intra-patch variation that we were able to measure. So first I want to show you this slide. This slide is going to be a template for the plots you're going to see on the following slide. They're all going to look like this, but they're going to be 4 of them, each for a specific metric. And the idea is that these green boxes denote a number that is good for ecological health, while the red ones indicate a value that is worse for the health of an ecological system. So for the number of large trees that we saw in Area C, as you can see, they're concentrated on the east and west sides of that north and south transect. And as far as the percent canopy covered goes, you can see that it follows a gradient along that north transect from west to east. And on the south transect, it's pretty low, except for over here on the east side, it gets a lot higher. As far as invasive species abundance and native cover diversity, they're both pretty spotty. There's only one spot on each of those transects where those numbers were particularly healthy. And this is important because if you wanted to, say, develop that was going to encroach on Area C somewhere, and you wanted to care a lot about native cover diversity, for example, you could take a look at a figure like this and say, well, native cover diversity is in its best condition over here on the west sides of the plot. So it's probably okay if we develop on the east side.
So stepping back away from the intra-patch variable and just variability and trying to understand Area C as a whole, we applied the metric of the Floristic Quality Index. And so every single species that we measured in Area C has a number from 0 to 10. And this is called the coefficient of conservatism. And these are values that are designated by botanists in New Jersey. And 0 denotes a plant that can grow in any disturbed area, whereas 10 is a plant that needs to grow in a very undisturbed areas and only grows in its natural ecosystems. And so we found over 24 native species in Area C alone, and we had a mean coefficient of conservatism of around 4, which yields a Floristic Quality Index of around 20. So how good is that? So according to the U.S. Fish and Wildlife Service, a score of 20 to 35 indicates a region of high vegetation quality. And also in terms of thinking about Area C as a whole, based off of our large tree surveys, we calculated that there were over 6.3 times more large trees in Area C if you extrapolate from the densities that we found. And so just illustrate over here on the right, the green dots are the projected number of large trees based off of a facilities survey of the area, and the black dots are what we extrapolated from our density. So we spent a lot of time in the field doing field work, which was very time intensive. But the question is, how do we take those time intensive field work surveys and turn them into a long-term monitoring system? So one thing that we're going to explain some more of is the implementation of using more remote sensing instead of having to spend time in the field. So what a long-term monitoring plan could look like is pictured here. So obviously it's important to first establish a baseline, and maybe this could be done every 10 years to recalibrate it. And that would require intensive field work, intensive time, but not very much money involved. The other options, using remote sensing, require more money but are much cheaper and can be done on a shorter time frame to understand the variability within those 10 years. So one thing that could be done, which would be a good first step, would be to start taking images of campus that are orthorectified leaf-on images. Currently, there are orthorectified leaf-off images, but all the leaf-on photography are just spot shots, which are sort of lower quality. And it would be also great to add a near-infrared camera to the orthorectified images to understand the vegetation health. These can be done maybe once every year, and as well as EB drone flights. So the Geoscience Department owns a few EB drones, and they could be used to target specific forest patches on a monthly timescale during the growing season of vegetation. These would be relatively low cost and would give high quality imagery. LiDAR, as I explained before, is a great metric to understand forest structure. It would need to be done much less frequently since it's not giving you any specific visual vegetation metrics, but it's more showing you the structure of the forest. So maybe every 5 years for this more expensive metric.
Thank you guys so much for bearing with us. We're going to go ahead and bring our presentation to a close here and just emphasize a few takeaways from the presentation as a whole. So first of all, this idea of transparent, reproducible methodologies, you've seen that coming up several times in the last 35 or 40 minutes. So I just want to touch on that briefly again and explain the importance of that in terms of monitoring these forest systems and campus as a whole in response to the developments that have been proposed in the recent campus plan, as well as the ability to accurately compare accurately compare areas of campus against each other when it comes to making more developments in the future. Also, making the historical data sets that are already owned by Princeton University more readily available, especially to students, would make this process significantly easier for us and also give it a lot more potential for further interaction with the students. And finally, these long-term monitoring systems that we've been talking about mostly recently, some of these would be super easy to install, like the ruler on the side of a bridge that was mentioned by the Lake Carnegie team in order to get a baseline for the water depth of Lake Carnegie. Also, rainfall gauges around campus, stream flow rates, these are things that wouldn't be that difficult to get and would really help in having continuous data sets for monitoring, as well as establishing baselines against which to compare further data in future studies. And finally, these are great opportunities to interact with students in the natural sciences. However, this is not the only area of the student body that you'll be able to interact with.
So yeah, this presentation was all around how natural science students can get involved with campus. However, I believe that students from the arts to engineering majors can get involved with campus and have more of an investment in their campus if there's some work that's put in to try and engage those students. Obviously, it's not something that is feasible for students to step in right now and just start engaging with the facilities to start implementing their ideas on campus. It is something that's going to need to take some investment from the Office of Sustainability, from facilities, to try and engage these students. But I feel like that this could be a very valuable investment because all of these students have valuable skills, valuable innovative ideas, and they could really start to have and feel an ownership of their campus and of their land they're working and learning on if they're allowed to sort of get more engaged with the process of managing our campus. Thank you. Thank you for coming. Can we get the AEI team up here and see if we have any questions?